DNA replication begins at a specific location called the origin of replication. Helicase comes to the origin of replication and unwinds the DNA and breaks the weak hydrogen bonds connecting the nitrogen bases. As the DNA unwinds, single-strand binding proteins attach to both sides to stabilize and keep them from reattaching.
DNA replicates in a 5' to 3' direction. The leading strand is oriented to grow at the 3' end when the fork is opened. Primase is the enzyme that builds the RNA primer, which is a short strand of RNA nucleotides. The primase builds the RNA primer at the start of replication, so dNTPs are capable of being added. After these steps, the main enzyme in DNA replication, known as DNA polymerase III, is initiated. DNA polymerase adds dNTPs to the 3' end of the primer. The energy needed for DNA polymerase III to create the phosphodiester linkage is produced by the removal of extra phosphates.
The ligand strand begins to replicate at the 5' end and moves towards the 3' end away from the replication fork. It is synthesized discontinuously as a series of short segments called Okazaki fragments. The process begins with primase attaching RNA primer to the ligand strand. DNA polymerase III comes in and uses the RNA primer to add complementary nucleotides in one fragment until it reaches the previous RNA primer. DNA polymerase III is then replaced by DNA polymerase I, which removes RNA and replaces it with DNA. DNA ligase then attaches to form phosphodiester bonds, and then the process repeats.